but now I'm going to introduce our speaker. So today we are privileged to have Craig Newschaffer with us speaking about autism and the environment. Craig Newschaffer is a professor in the Department of Epidemiology and Biostatistics, as well as the Dean for Research at Drexel University Dornsife School of Public Health in Philadelphia. Dr. Newschaffer is also the founding director of the A.J. Drexel Autism Institute. The Institute is the first autism research center to focus on public health science, including programs in modifiable autism risk factors, early detection and intervention, as well as life course outcomes. Over his career, Dr. Newschaffer has been the lead investigator on many federally funded autism studies, including a National Institutes of Health Autism Center of Excellence study focusing on prenatal autism risk factors. It is our extreme pleasure to have Dr. Newschaffer with us today, and so without further ado, please take it away.
Thank you very much, Amy, and thanks to the folks at Simons and Ian and Spark for hosting me. Let's make sure I'm in the driver's seat here. All right, I think I successfully advanced the slides. That's a good start. So the title of my presentation is 4 Things You Should Know About Environmental Autism Risk Factors. And this slide shows the outline of what I'm going to try to get through this afternoon. First, I'm going to talk a little bit about this term risk factor, try to describe a little bit what it is, what it means, how we think about it. And then I'll do the same thing for another commonly tossed around term, environment. And I'll focus a little bit on what environment means in the context of causation of health outcomes. Then I'll work through my 4 things, the 4 points I wanted to convey. The first, that environmental risk factors do contribute to causing autism. This is something we know. That environmental risk factors are challenging for scientists to discover and confirm. 3, that the amount of increased risk any one environmental risk factor conveys at the individual level is likely quite small. However, the overall impact on the population can be quite large. So this contrast between thinking about risk factors as an individual or a family member of an individual, prospective parent, thinking about risk factors in that context can be quite different than thinking about risk factors from the context of maximizing benefit for an entire population. And I'll try to underscore that point throughout the talk. Then I'll close by just going through a short discussion of some particular classes of environmental risk factors that have some supporting evidence and that are sort of under most active study right now. So I'm not going to go through, this talk really will be more conceptual. I'm not going to go through a laundry list of candidate environmental autism risk factors and talk about evidence pro and evidence con. I'm going to focus on what I think are some really important things for you all to think about as you're learning about environmental risk factors, as you see reports around those in either the technical literature or even the general press. And then we'll try to leave some time for questions at the end. And I will watch the clock carefully to make sure I don't talk too long. All right, so let's begin with what is a risk factor? So a risk factor is something that affects the chance of an outcome, like a health condition, but it could be any other outcome. So when we talk about chance, what we're really talking about is probability. And so probability is a math term, and yes, when we think about risk factors, there is a little bit of math thinking involved. That can be challenging to some, and there can be some room for some misinterpretation of some of the basic math underlying the concept of risk and risk factors. So here's sort of a risk estimate that we encounter every day on our smartphones. If we have a weather app, we see a risk estimate that there's a 30% chance of rain, in this case at 11 a.m. in New York. And this is a pretty simple piece of information, but it's worth thinking a little bit about what that means. So when we contemplate what 30% chance of rain means, we have to first think about what is this probability applying to? And what it's really applying to, it's applying to the set of hours where the factors in a weather prediction model suggest that the conditions are what they think they're going to be observing at 11 a.m. on this particular day. So there's a whole set of infinite days that will have those conditions, and on a day with those conditions, we expect to see rain for 30% of the time, or 3 out of 10 of those days. Now, the other thing to realize about these kinds of risk estimates for weather forecast, a chance of rain estimate, is that New York here, what's this applying to? It's applying to New York, and New York is a pretty big city, and these particular risk prediction estimates also affect the size of an area. So when a weatherman is making this prediction, or the weather forecasters that work for Apple are making this prediction, they're thinking about the totality of the space of New York City. And if they are 100% sure that rain is going to fall based on their model in one half of the city, but in the other half of the city they believe based on their model there's a 0% chance of rain, what they would report in this kind of a statistic is actually that overall for New York City there's a 50% chance of rain. So this, I'm using air quotes, you can't see me, but I'm using air quotes to say this simple estimate of risk is actually more complicated than we might think it is, and there's a reasonable amount of uncertainty involved in it. We know off the top that it means that in 3 out of 10 days like this, rain may fall, but there's also going to be some dependency on where we are in New York City as to whether or not we're going to get wet at 11 a.m. on this particular day. So there's a lot of uncertainty in this estimate for the individual to think about when they're thinking about pulling an umbrella out or not that morning. And the kinds of probabilities that go into the study of risk factors, where we contrast the probability of an outcome occurring for individuals who have a candidate risk factor to the probability of an outcome occurring in individuals who do not have that risk factor. The probabilities that we use are very much like this. And sometimes they even have more uncertainty, because in research studies these probabilities come from a sample. For weather forecasts, these weather models are able to basically predict conditions for every day and every hour out of the year in a particular location. So they're actually observing the universe, and they still have a fair amount of uncertainty, as we just discussed. In research, we've got all that kind of uncertainty, plus we have the extra added uncertainty that comes with just studying a sample and not being able to observe the entire universe. So a lot of uncertainty in these risk estimates that go into understanding risk factors that's going to have an implication on individuals making decisions. But this kind of information can still generate evidence that is very helpful to scientists and is very helpful to individuals who are thinking about health and health outcomes at a population level, that we'll get to in a minute. All right. So what about the environment? So in the context of thinking about cause of health outcomes, when we use the term environment, what are we talking about? Well, we're basically talking about everything that is not heritable genetic determinants. And that is a huge everything. It's dietary factors, it's lifestyle, it's your surrounding, it's your experiences, stressful events, happy events, exercise, built environment. It's all of these factors together constitute the environment. A lot of times when you think about or you hear the term environmental risk factors, you're sort of thinking about sort of hazardous environmental exposures, things that are a little bit fearful to think about, like industrial chemicals. But it's not only that, it's this whole constellation of experiences outside of heritable genetics. And the fact that this space is so huge is a point that I'm going to return to in a little bit. The other thing of note about the environment is that environmental factors are often changeable. They can be hard to change. It's hard for us to change our individual behavior. It can be hard for us through public policy processes to change the natural environment or the physical environment, but it can be done. When we think about heritable genetics, changeability is much more of a challenge. And I think if you read the news, we all know that we're on the cusp of a new era in genetics with the advent of CRISPR, the technology that actually allows us to edit genes. But we're at very, very early days there, and it's unclear what the therapeutic potential of gene editing is going to be. But we do know that we can change behaviors, we can change the environment. So that's very important when thinking about this class of risk factors. All right, so now that's the background. Now I'm going to transition and talk a little bit about my first major point I want to emphasize, that environmental risk factors do contribute to causing autism, and this is something that we know. So, on the other hand, we also know for sure that heritable genetics contributes a lot to autism. The Spark Initiative, as you know, since you're a part of this community, has an emphasis on genetic risk factors, and they're very important in thinking about the cause of autism. But the question is there also room for factors in the environment to also be contributing to cases of autism? So this slide has a lot of numbers on it. It's the most numbers I have on a visual image today. I'm not going to go through them, but it's there just to help remind me to talk a little bit about the first class of evidence that we have that supports the idea that environmental factors contribute to autism, and that comes from family studies. And when we conduct family studies in autism, and maybe you've heard about them in previous webinars or other fora, what we're doing is we're contrasting the recurrence of conditions among family members who have different levels of relatedness. So basically, if we compare monozygotic twins, they have a high degree of genetic similarity, and if we look at twin pairs in which one individual twin has a condition, like autism, the recurrence is whether or not the other twin also has autism, and we contrast that rate of recurrence that we see in those monozygotic twins to other kinds of family members, like dizygotic twins, or cousins, or non-twin siblings. And when we do this, we estimate something called heritability, which is often interpreted as the amount to which heritable genetics contribute to a condition. So, again, my table shows some results from twin studies over the years. The yellow highlighted twin studies are the more modern era studies. But basically, when you put this evidence together, the heritability estimates from these twin studies suggest that about 50 to 60%, and there are some estimates some studies that suggest even more, 50 to 60%, maybe more, of autism is attributable to heritable genetics. And then other kinds of family studies, some that have been published quite recently, have suggested even higher heritability estimates, estimates that are above 80%. So, a couple of points right here. So, while that underscores the idea that genetics is important in autism, it also has a considerable proportion of causation estimated to be due to non-genetic or environmental factors. Okay, so that's the starting point from this kind of data. So there's a little bit of contentiousness, I think, when we talk about genetic and environmental contributors to a condition. And this doesn't only apply to autism, and it's a little misplaced, but I think it exists for a couple of reasons. I mean, I think that some of it's our fault, the research community's fault. The kind of scientists who do genetics research are different from the scientists who do research on environmental risk factors, and we're all out there trying to get our research grants, and so we tend to emphasize what we know and study in order to compete for scarce resources, and that's undeniable. So, but there's also some differences in sort of the outlook, I think, of the genetics research community and the environmental research community that flow from history. So I think that in genetics, it's been a field that was really sort of defined and built on studying single gene disorders, conditions like Huntington's disease, cystic fibrosis, hemophilia, and others, where a single mutation in almost all cases, causes a health outcome. And so this gets you into sort of a deterministic mindset. Now, genetics researchers today who study more complex conditions like autism or diabetes or breast cancer clearly are of a mind that causation is complicated and the genetics are very complicated, and fully realize that the genetics that they're studying are not of that type of mechanism where you have one mutation and you see a health outcome. But that history is still there. On the environmental side, the history is kind of the opposite. Think about sort of the most prominent environmental risk factor for serious health outcome, cigarette smoking and lung cancer. Powerful risk factor, right? The risk of lung cancer is 10 to 20-fold higher in smokers than it is non-smokers. But from the beginning of the course of the study of smoking and lung cancer, we knew that 10 to 15% of all lung cancers arise from non-smokers. So in fact, lung cancer in non-smokers would be a top 10 most common cancer if we counted it as a separate category. So a lot of lung cancer is due to other mechanisms, and so the environmental contribution of even this most strong environmental risk factor, has its limits. So environmental risk factor researchers have always been sort of more accepting of a lack of simple causal mechanisms and complexity. And I think for a condition like autism, even though there's a large genetic component, this kind of complexity is very much the situation that we find ourselves. So let's use a cartoon to illustrate some of this causal complexity. And so I'm going to go through a couple of these different pie charts. These are called causal pies. And the way to think about these is a circle like this represents the constellation of causes that are necessary to lead to a health outcome. So in this particular cartoon, there are 8 different component causes that have to come together to bring about the health outcome. And it's also useful if you want to ask yourself the question, well, what do we mean when we mean a cause? These causal pies are quite helpful, because what it means is if you were to, in this situation, for this mechanism, if you were to take away one of these risk factors, one of these causal contributors, pie slice V, for example, then you would no longer see the health outcome in the individual who had the other 7 component causes. So when you take away a factor, you don't see the health outcome. That's one definition of cause that's very useful. So single factor causation, the causal pie would look something like this, right? There's one factor needed. I've given it G, just the idea that this might be a monogenetic condition where all that's needed is a single genetic mutation. And it is sufficient in and of itself to bring about the health outcome that's in question here. Now let's look at this next causal pie. So this is still a fairly simple causal pie. All I've done is built it out so that there's now 2 components that are needed to bring about a health outcome. G is a gene, and E represents some environmental risk factor. So both of these are needed, and this is slightly more complicated, and this is a situation that we call gene-environment interaction. And there are a couple of things I want to emphasize about gene-environment interaction. The first point I want to make is to establish that this is something that really does happen quite a bit. So if we think about the condition PKU, phenylketonuria, right? Many of you have heard about this. It's an inborn error of metabolism. We screen all newborn babies for it. It is due to a mutation of one gene. And if you have that mutation, you can't metabolize a common amino acid in food and protein. And the results can be fairly serious. There's serious skin conditions, and there's also cognitive impairment that's associated with PKU. And we think of PKU as a genetic condition, but the fact is it really is a gene-environment interaction, because if you have that mutation and you can't metabolize phenylalanine, if you alter the environment, you don't expose an individual who has that mutation to that amino acid. You change their diet, you give them a low-protein diet, they will not have the clinical manifestations of PKU. So really it is the interaction between the genetic mutation and the environmental exposure, the existence of phenylalanine in the diet, that brings about the clinical health outcome. So it's an example of gene-environment interaction, and those exist all the time and probably exist in many more complicated forms, for sure. The other point I want to make with this causal pie has to do with this idea of the relative contributions of genetic factors and environmental factors to a health outcome. So let's think about this pie this way. Let's say that this is the only way that a particular health outcome can come about. This is the only mechanism. And remember, we talked about if we take away one of these causal components, we don't realize the health outcome. So for this condition, a question I could ask is how much of the condition is genetic? So if we look at the pie and say, all right, well, if we could somehow remove the gene through the magic of CRISPR or through generations of evolution, if the gene is gone, the condition cannot exist. So 100% of the condition is due to heritable genetics. But now let's ask the other question. What if we were able to eliminate the environmental risk factor? Well, if we eliminate the E, the criteria of the pie aren't met and the condition doesn't exist. So again, 100% of the condition is due to the environment. So right there, the fact that the causal contributors add up to more than 100% is something that is very, very challenging for the business of estimating how much of a condition is genetic versus how much of a condition is environmental. And in fact, those heritability estimates that I talked about when I showed you that table of all the twin study data, they overestimate heritability because they assume no gene-environment interaction. And that's, in my view, a very naive assumption. So those estimates that we had, whether you're in the 50-60% range or you're holding on to a higher estimate of heritability for autism from some of the more recent family studies, you have to realize that they're likely overestimated. And the room for environmental factors to be influencing the outcome in question, in this case autism, is probably substantially larger than what those heritability studies put forth. All right, so that's my first sort of set of evidence supporting this idea that environmental risk factors do in fact contribute to autism. The other thing I wanted to touch on briefly before moving on is that we do have some proof of principle examples of environmental exposures that cause autism. So this is a citation for a study, a very famous study that involved a group of researchers who were going back to re-examine the records of 85 individuals who were exposed to thalidomide in the 1950s, whose mothers took thalidomide while pregnant with them in the 1950s. Thalidomide it's a powerful teratogen. It causes birth defects. We didn't know that in the 50s, and it was prescribed as an anti-nausea agent. And so there was a large spike of individuals who were exposed prenatally to thalidomide, and it caused horrible birth defects, and so it was detected fairly quickly and it was no longer used for that purpose. But these researchers were going back and re-looking at records for 85 exposed cases. And they found that, and they were doing this in, the paper was published in 1994, but they were doing this in the late 80s. So when they did that, they found that there were 4 cases of autism in this series of 85 thalidomide-exposed individuals, which was a very high percentage. I mean, that was about 5%, which was wildly high for the late 1980s when autism prevalence was thought to be well below 1%. And what they also noticed, though, was that every one of those autism cases in the series, the moms were exposed to thalidomide during the same gestational age window of 20 to 24 days. So there's a very tight correspondence of timing of exposure prenatally to the occurrence of autism in this case series. And it's pretty strong evidence that thalidomide, in addition to being a teratogen and causing physical malformations, also causes autism. So we have other examples like this, rare exposures that do cause autism. So you put that on top of what comes out of the family studies, and I think it's pretty clear that environmental risk factors are contributing to causing autism. Okay, so point 2. Basically, why don't we know more? environmental risk factors are challenging for scientists to discover and confirm. So part of the challenge to uncovering and revealing those risk factors has to do with the scope, this huge space that we have to work in. And if we think about all the domains of environment, from physical, psychological stress to diet to lifestyle to classic environmental chemical exposures, they're all wildly complex and large. I'll focus for a second, not because I necessarily believe that it's ultimately going to be more important, but because I have some illustrative numbers I can share with you. I'll focus on the domain of environmental chemicals for a minute, just to illustrate how complicated this space is. So this is sort of an iceberg analogy that's often used to sort of explain the vastness of the landscape of environmental chemicals. So at the tip of the iceberg, there are 8 to 10 chemicals manufactured, synthesized by humans, not natural creations, right? Artificial creations, that are proven developmental neurotoxicants. We know that they adversely affect the developing brain, for sure. It's a very small list. Now, we can detect in blood and urine of all Americans through biomonitoring surveillance, including pregnant women, more than 200 now synthesized chemicals. So the proportion that's been proven to be neurodevelopmental toxicants out of all those that we can detect in every American is quite small. But that doesn't mean that we have assiduously studied all 200 of those chemicals. Probably less than 20% of those chemicals have been adequately screened for neurodevelopmental effects. So there's a large number, more than 100 of these commonly detected chemicals that we haven't adequately screened for neurodevelopmental effects. Those 200 chemicals are only a fraction of what is a much larger number of chemicals that are produced at very high volumes and enter the environment at very high volumes. And high volumes classification means that more than 1 million pounds per year of these chemicals are produced. And there are more than 3,000 of those. And those 3,000 high volume chemicals are part of a chemical universe that comprises more than 80,000 synthesized chemicals. So while we've learned some things about exogenous chemicals that have neurodevelopmental effects, there is so much more that we don't yet know than what we do know. And figuring out strategies to effectively query this large range of environmental chemical exposures, and then also query the large domains of other environmental classes of exposures, is a real challenge for researchers like myself and my colleagues. Pivoting back to the genetic side of things for a minute. Part of the reason why there's so much excitement in the world of genetics, and rightfully so, is that the molecular biology revolution has brought forth the technology with which the entire genomic landscape can be interrogated. So we now, through techniques that began with GWAS technologies, and we're now into genetic sequencing, we can essentially, from a sample of anybody's DNA, and again, if you're following along Spark webinars, you've probably heard about this kind of thing, or you will soon. We can basically measure every base pair in an individual's genetic code. There are 3 billion base pairs. There are 25,000 genes. We can characterize all 25,000 of those genes for an individual, and we can use new types of discovery-based statistical approaches to analyze the full sweep of the genetic background. Now, I'm simplifying a little bit because the more we learn about genomics, the more we learn that there are complicating wrinkles besides just the alphabet that is your genetic code. But the technology has come a long way, and we can get our arms around the genomic background of any individual. If we now pivoting back and think about environmental exposure, we don't yet have sort of a comparable approach to doing this. Our science is based on studying 1 or 2 or 3 exposures one at a time. We can't comprehensively get our arms around the history of environmental exposures that any one individual has faced, whether we're talking about the prenatal period or early childhood or whenever. There is a lot of interest, though, in trying to sort of change this. And so this term that you may or may not see, the exposome, is something that is coming into wider use. And really, when we talk about the exposome, we're talking about trying to do something that we can do with genetics on the side of environmental exposures. Can we, in a biologic sample, whether it's blood, we work here at our research center with other biologic samples like shed deciduous baby teeth. We work with meconium, which is baby's first bowel movement. We work with these samples and try to see if we can accurately measure signatures of a huge range of exposures that individuals may have come in contact with. And this work, this exposomics work, is at the very, very early days. And it is one of the reasons, the challenge of the huge sweep of the environmental landscape and our inability to sort of wrap our arms around it from a measurement perspective, that is a bit of an impediment to doing research on the discovery of environmental risk factors for autism. All right, so now, 2 points down, 2 points to go. Running a little behind on time, but I think I can bring it in with plenty of time for questions. So, shifting a little bit, we were talking a little bit more about some of the research challenges. The question of what's the evidence like supporting environmental risk factors, supporting the idea that they have a role in autism. We talked about that. We talked about some of the challenges that we face when we try to do discovery research on environmental risk factors. But now I want to shift and talk a little bit more about interpretation. And the fact that the amount of increased risk that any one environmental risk factor conveys at the individual level is likely quite small. And what this means for individuals who are contemplating their risk or the family member's risk or the risk of a child that they might be thinking about having someday. So I'll use as an example some data from a paper that we published a few years ago on prenatal exposure to selective serotonin reuptake inhibitors, one of the more commonly prescribed medications for depression. And this is in utero exposure to the drug. So women who have severe depression may continue to take their medications while they're pregnant. Severe depression is a major health challenge in and of itself, and being actively depressed while you're pregnant is not good for the health of your baby. So decisions are often made to continue medical management with these kinds of drugs while you're pregnant. And so we've done some work looking at the risk that exposure to that kind of medication conveys with respect to autism. And the way this risk is presented is as a risk ratio. So it is actually a ratio of the probabilities that autism occurs in children whose mothers were exposed to SSRIs in pregnancy, divided by the probability that autism occurs in children whose mothers were not exposed. And that ratio measure is 1.9. It's nearly 2. So that statistic can lead to statements, and these are correct statements, that risk was almost doubled in moms who took SSRIs while they were pregnant. Risk of autism in their children was almost doubled. And that is a scary sounding statistic. But what we need to do when we think about the results of this study at the individual level is think about the difference in risk, not the ratio. So if we just assume for a second that the risk in this study for the babies whose moms did not take SSRIs in pregnancy is about the population prevalence of autism, which is 1.5%. So I put that number here in the denominator of the ratio, right? And then that means that the absolute risk to the children of the moms who did take the SSRIs in pregnancy to generate this relative risk estimate of 1.9 would be 2.7%. So if we think about the contrast of 2.7% and 1.5% as an absolute difference, that difference is pretty small. It's a 1.2% absolute difference in risk. So based on this study, if it's correct, and I'm not saying it's correct in and of itself, this is an active area of research, but if these estimates are correct, what this is saying is that if you take SSRIs in pregnancy, the absolute increase in risk will be 1.2%. So your child will have a 2.7% chance of having autism compared to the baseline 1.5% chance in the population. So when you're considering suspending the use of what can be an incredibly helpful medication that affects symptoms that also influence pregnancy outcomes, this difference, this small difference in absolute risk, might be one that moms are certainly willing to take. And the risk does not seem as scary as it does when it's presented as a ratio, when we say the risk is doubled. Right? Now, so the message there, before I transition, I just want to recap. So the message there is that when you're thinking about the impact of environmental risk factors at the individual level, it's the risk difference, the absolute change in risk that folks are going to want to think about. At the same time, though, we also want to think about the effect of environmental exposures in the whole population. Sure, it's important to think about them for an individual who has to make decisions about whether or not they want to continue with an exposure or try to avoid an exposure. But it's also important to think about these risks from the population perspective. And in fact, these risks, even risks that are small at the individual level, can have large population health impacts. And to make this point, I'm going to use data from another study that we did. This time, this was a systematic review, so it wasn't just the results of one study. It was a review of a bunch of studies that have been done on air pollution and autism risk. In particular, air pollution, again, during the prenatal period when the brain is developing. So what's the effect of exposure to air pollution during the prenatal period on the risk of autism in the children born to moms with different exposure levels? All right. So here we presented a summary of the research, and we also presented a ratio-based measure of association. So we said that the risk was more than doubled. The relative risk, or the risk ratio, was 2.3 for each standard deviation increase of exposure to one kind of pollutant, which is PM2.5. So let me unpack this a little bit. So PM2.5 are small particles. So it's particles in the air that are less than 2.5 microns in diameter. That's less than a third of the diameter of a human hair. So these are tiny, tiny particles. Now, standard deviation, another math term. What am I talking about when I talk about a standard deviation? Well, if you think about a distribution of exposure in a population, right? How many people have high exposure, how many people have low exposure, and how does that distribute? Think about a bell curve, and I'll show you a bell curve in a minute if you can't remember from high school statistics what the bell curve looked like. It's that symmetrical bell-shaped distribution where most of the people are in the middle and fewer people are out at the extremes. So standard deviation is the way we measure how wide that bell curve is, how much variability there is. But basically, if we look at standard deviation as a unit, two-thirds of the distribution, two-thirds of the middle of that distribution, the majority of folks are between -1 and +1 standard deviation of the mean of the distribution. Right? And then if you add on another standard deviation in either direction, higher exposure or lower exposure, you're capturing, gosh, a little over 95% of the entire population. And if you go out 3 standard deviation units in either direction, you've got everybody. Now again, I'll show you a picture in a second to remind you of this. But let's before we get to that, let's take a look at this ratio-based measure of association, sort of the same way we did for the SSRI example, its impact on individual risk. So I do the same thing. I break down the relative risk into 2 numbers, one representing the risk in the comparison group, and here I'm assuming that that group of people that 1/3 of the population whose one standard deviation below the mean level of air pollution of PM 2.5 air pollution exposure, that their prevalence is about the average, 1.5%. And that a one standard deviation increase, which takes you one standard deviation above the mean, the average air pollution exposure level, to get this ratio measure of 2.3, that means that the autism risk in that group is about 3.5%. So thinking about it as an absolute difference of 3.5% to 1.5%, again, we're talking about a relatively small absolute difference in risk. So the idea of picking up your entire household and moving from an area where your PM 2.5 exposure might be in the one standard deviation above the mean range to another area where it's one standard deviation below, you'd only be reducing the absolute risk of autism occurrence by 2%, a small amount. So the individual impact of this exposure seems to be fairly modest. But there's that bell curve. And let's think for a second about the population impact. So this curve that I alluded to before, this represents basically everybody in the population. This is a distribution of everyone's air pollution exposure, so that individuals who are out here have very high exposure, individuals down here have very low, and there's that 2/3 group sort of clustered around the middle, right? So that's a standard bell curve. So now that estimate, that 2.3 relative risk, how does that translate throughout the entire population? Before, I showed you a contrast of folks who are here to folks who are here. So if we think about how that autism risk estimate translates throughout the entire population, it's this kind of a risk function, right? The individuals who are at very low levels have lower expected autism occurrence, 0.1%, and the individuals who are in this highest, right, in this 3 standard deviation units above the mean, have an alarmingly high autism occurrence rate, a prevalence of 8.5%. So the first thing that you might think is, oh my gosh, this is alarming. Let's focus on this group of individuals who are very highly exposed. So what could we do? If we're public health practitioners, we could try to think of a way to reduce the exposure of these individuals who are way out here in this third distribution class and move them down into the point where they're between 1 and 2 standard deviations of exposure. So hypothetically, snapping my fingers, if we could do that, what would be the consequences? How much autism in the population would we reduce by taking that public health action? Well, by doing that, and I see I've just moved them over, I've snapped my fingers and I've moved their exposure over. By doing that, the impact on autism prevalence in the population would be relatively small. The overall prevalence would go down to 1.4%. So it would go down from that population average of 1.5% that we observe in real data. If we could make this magic intervention, it would go down to 1.4%. That's only a 3% reduction in cases overall. So, I mean, very important for the folks who we've moved, but the population impact is not that large. But what if through public policy or public health action there was a way that we could move everybody's exposure down 1 standard deviation unit? So we shift everybody down. We lower everybody's exposure. The individuals who are out here at the highest exposure and therefore have the highest risk of autism, but also individuals down here who are at relatively lower risk of autism to begin with, but we improve their exposure by reducing the amount of PM 2.5 that they breathe. So you go ahead and do that. And if we do that, now we see a tremendous impact, right? We see that the prevalence of ASD has moved from 1.5% to 0.8%, a 47% reduction in ASD. Now, I'm not saying that I think that we can cut autism in half by reducing PM 2.5 exposure. I think there's a lot we need to learn about the relationship between air pollution and autism for sure. But what I want this to illustrate is that this environmental risk factor, that at an individual level may not have been the most impactful thing. I don't want to move my household for a 2% decrease in absolute risk. From a population perspective, policies that would reduce air pollution exposure dramatically could have a huge impact. And this is why decision-making about these kinds of exposures, at least in my view, I think appropriately has to happen at the societal, governmental, regulatory level, because this is where we see the potential for the greatest returns. All right, I'm getting a little tight on time. I've got a couple of slides I'll go through quickly, and then I'll open it up for questions. So, specific environmental risk factors that have some supporting evidence that are under study now. We can talk more about these in the Q&A period if you want. But basically, there are pre-perinatal and neonatal complications that have been linked to increased autism risk, including low birth weight and prenatal infection. And then there are a large list of complications here that have less supportive evidence that we're interested in learning more about. We know that there's a relationship between increased parental age and autism risk. I mentioned an example of prenatal medication use, and there are other classes of medication that are of concern and under study. Short interpregnancy interval, short time between pregnancies has been shown and replicated to increase autism risk. Nutritional factors, potentially important. Folate, folate supplementation, taking prenatal vitamins with folate, has been shown in most studies to reduce the risk of autism, although there are studies that haven't shown any effect. Prenatal air pollution I've talked about, and then the whole class of chemicals of particular interest are chemicals that disrupt the endocrine system. Now, we can talk more about any of these, but what I do want to say is that based on the evidence that's out there for all of these, sort of the magnitude of the effects are of the same ilk of what I showed you, where the individual effects, the risk difference, what an individual would think about if they were thinking about avoiding or changing behavior, the risk difference is relatively small, but the relative effects can be relatively high, and if these are common exposures, the population impact can be fairly substantial. So if you want to read more about these, this is an academic article, but it's written in very plain English. It's in a journal that most public libraries have. We go through most of those classes of environmental risk factors, and it's something that you can find fairly easily. If you're really interested in individual decision making, you're worried about, you're contemplating a pregnancy, and you really want to dig deeper into these smaller risks, but you want to know what's what with that, this is an excellent resource, the Program on Reproductive Health and the Environment at UCSF. If you go to their website, they have a series called All That Matters. You can find it on their homepage. It's in English and Spanish, and they've got very nicely written materials that talk about the individual level risks associated with things like occupational exposures and diet and exogenous chemicals, things that expecting moms are going to come across in their daily life and things that they may be worried about. So that's a resource that you can utilize. So with that, I ran a little long, but we still have 9 minutes. I can take some questions.
Great. Thank you so much, Dr. Newschaffer. We have a bunch of questions that have come in, so I'm going to try and run through as many as I can, and then we will make sure we address some of these through our website or social. So one comes from, I believe, a parent with 2 children, both of them with autism, but one of them is more severe. So if both of them have the same genes and the same and were exposed to the same environment, her question is, what are the things that make one more severe than the other?
Yeah, that is a fantastic question, and there's active research in that very area. So there are a couple of things that could be considered. One is the postnatal environment, where things start to differ, but if they're raised in the same family, same general habit, same general lifestyle, there's not going to be tremendous differences there. But the other thing that we're very much interested in are factors called epigenetic factors, and I don't have enough time to go into it, but it might be a great topic for one of your future Spark talks. In addition to the genetic code, the A's, the G's, the T's, the C's that are in our DNA, and that are, for monozygotic twins, identical, there are other chemicals that actually physically adhere, for example, to DNA that can vary and can vary across closely related individuals and even vary across monozygotic twins. And the thinking is that perhaps those epigenetic factors might be the thing that brings about differences in something like severity. There are even instances of monozygotic identical twins where one twin will have autism and the other twin will be fully typically developing. And so looking towards these epigenetic factors is something that is very much of interest to solve those riddles.
Great, thank you. Another question is a general one about a father's exposure to environmental factors. So can you talk a little bit about that, about what role the dad may play?
Yeah, well, it is a great question, and we have a not too proud history of focusing perhaps too much on moms when we think about autism causation, and we do need to think about exposures to dad. And it's just a coincidence, but the epigenetic word will come up again in my answer here. So fathers. Women are born with a full complement of eggs. Fathers manufacture sperm in real time, and in fact, the things that dad are exposed to can influence. While they can't change again that DNA code, the A's, the T's, the C's, the G's. What they can do is they can have an effect on those other chemical factors that adhere to the backbone of DNA. So it's possible that dad's environment prior to conception could influence epigenetic factors in the sperm and thus could affect risk to their children for autism and also for other conditions. Epigenetic mechanisms are suspected to be important in a range of complicated conditions.
Okay, thank you. So we got a lot of questions about specific factors, environmental factors, and whether or not they pose risk for ASD. So I'm going to run through a list, and if any stand out, let us know. So prenatal exposure to alcohol and marijuana is one. Screen time, I assume TV watching is another. Let's see. Traumatic birth effects. Let's see. And then one tricky one, AVMs, venous malformations in the brain, which I'm not sure what they are, but those are some other questions people had.
Okay. Yeah, I can talk about all those. So alcohol, cannabis, illicit substances. So there's actually fairly limited investigation of this, and we do talk about this in the article, but most of the studies have not suggested a particularly strong effect on autism risk. However, I do think that the research that has been done is a little antiquated, and I expect that in some studies that are underway now, there will be some attempts to replicate looks at alcohol and marijuana exposure in those populations, but for now the evidence is not there supporting a strong association. Screen time has not been well studied. It's supposed to be early life screen time, or I'm assuming as opposed to prenatal screen time. I'm not sure what the mechanism would be there, but has not been well studied. Most of my colleagues who are brain scientists, neuropsychologists, or brain scientists have a hard time thinking about the plausibility of a mechanism, about how, and I know that some have been proposed, but I'm just reflecting what really is just conversations I've had with colleagues, so it's not empirical evidence. The straight answer is there's not been a lot of research on it. But the reaction I get back from my colleagues who are brain scientists is that they can't think of a real plausible mechanism, so their enthusiasm for the hypothesis is fairly limited, but there has not been many studies. Traumatic birth events. So complicated pregnancies, birth complications was on my list, and I think it's something we do need to learn more about. Events that induced hypoxia, lack of oxygen to the brain, if you look at those studies individually, the evidence has been fairly mixed. Some studies have suggested increased risk, others not. However, if you step back and look at studies of any kind of birth complications, pretty much they almost always show some effect. Sometimes the specific complications that pop out are different, but I think we really do need to spend more time, and in fact this is an area I'm very interested in trying to figure out if that is real, and if it is real, what's the underlying mechanism. So that's something that is worth more study, and I think is potentially important in autism. As far as arteriovenous malformations, I don't know. I don't know the relationship of those. Those are congenital malformations of vessels in the brain, and I don't know of any work that's been done looking at those as autism risk factors.
Thank you so much, Craig. Okay, I'm going to ask one more question, and then we're going to close it out. Irene's going to close it out, but I do want to thank everybody for sending in so many questions. You can't see the list here, Craig, but there's many, and so obviously people are very interested in this topic. So thank you all very much, and we'll do our best to answer them later. The last question we wanted to ask is whether or not there's any, does the environment play a role at all in the higher male-to-female ratio we observe in autism?
Oh boy, great question. Unknown. Unknown. I feel like it could, and it is something that our group has actually started to look at, because you know, as you know, and this may be a topic of other talks that you have, you know, the reason, even though we've been aware of this disparity for decades upon decades, the exact reasons are still somewhat unclear to us, even with all the advances on the genetic side of things. There certainly are hormonally mediated mechanisms that are responsible for sexual dimorphisms, for differences between boys and girls, and there are environmental pathways that can act on those hormones. And so, for example, we have a study funded right now. We're analyzing the data that's looking at exposure to a chemical called triclosan, which is an antibacterial soap, an antibacterial agent that was used in hand sanitizers. It's in some toothpastes. And one of the things that we're potentially concerned about, and this is speculative, is that triclosan could affect hormones. It's believed to affect thyroid hormones, but we think it might also affect sex steroid hormone levels. Now, there's a lot of ifs there, and this is very early stage research, but that is just an example of how an environmental factor, if it does work through that pathway, could potentially be related to some of the sex ratio difference that we see. Is there any evidence supporting that particular hypothesis or any other? No, but it is something that is worth investigation.
Thank you so much. That's all the time that we have for today's webinar.